Hello, my name's Jonathan Coleman. I'm a postdoctoral research associate in Jerome Breen's lab at the Institute of Psychiatry, Psychology and Neuroscience at King's College London. And thank you for inviting me to talk about our paper, the title of which you can see displayed in the slide there. This is, of course, a journal club, and the paper is available on BioRxiv, and it's under review. So please, comments are very much appreciated. So first, I'll briefly mention a few key papers, which I will discuss in greater depth later. These are the SNEAKers intelligence meta-analysis published this year in Nature Genetics, Zabana et al., which is a genome-wide association study, or GWAS, including individuals with extremely high IQ, and Skeen and Brewer and colleagues, which describes a single-cell RNA-seq database of mouse brains, a method for identifying brain cell type expression specificity, and the application of that to schizophrenia GWAS results. As an aside, intelligence genomics has boomed with the release of the full genetic data from UK Biobank, and these three key publications have emerged since we put our paper online, and I will make some occasional references to these as well. So genomics and intelligence has followed a fairly typical path for a genetically complex trait, that is, one in which multiple variants across a range of effects are likely to contribute. Specifically, a gradual increase in sample size has identified regions of the genome and particular genes at genome-wide significance, although prior to this year there had been little replication and the robustness of these findings was in some doubt. To address that, SNEAKers and colleagues brought together a number of genomic studies that had assessed intelligence using various methods at different ages, focused on individuals of white Western European ancestry. And they built a meta-analysis of 78,000 people. The effect of that was they were able to identify considerably more significant genes and loci, and to reinforce some of what had previously been described. And this was the situation in which we undertook our study. So one of the key developments necessary in complex trait genetics is to be able to translate statistical correlations between DNA variants and observed traits into meaningful and actionable findings, whether biological or cultural. Well, we were lucky enough to be involved in a project developing such functional annotation. So the Karolinska Institute mouse superset is a single-cell RNA sequencing dataset of cell types in the mouse brain, and is the most extensive both in terms of the cells sequenced and the cell type assessed, currently available. So Skeen and Brewer and colleagues describes this dataset and demonstrates a method for testing the association between gene-wise association scores from GWAS and specificity of expression in cell types, although this actually extends to any tissue. And here I'll show the depth and breadth of the KI superset. So the set is divided into 24 level 1 cell types, which are these broad groupings you can see on the left, and 149 more specific cell types, which are termed the level 2 cell types, and are subdivisions of level 1. The association method has its basis in the cell type enrichment score and uses unique molecular identifiers which tag DNA sequencing reads. For the enrichment score is the mean count of those unique molecular identifiers in each cell type divided by the sum mean of these identifiers across all cell types. When you do that for all genes, you get a distribution of gene specificity scores in a given cell type, such as this one. So genes on the left are relatively underexpressed in this cell type, and genes on the right are highly expressed. The shape of the distribution varies between different cell types. While this is an example distribution, not all distributions will look like this. The association can then be tested in different ways, and the method used both in our paper and in Skeen and Brewer uses two alternatives. On the left, the distribution is binned into 41 bins, and a linear regression is carried out between bin membership and gene-wise association statistic, a GWAS. On the right, the upper decile of the distribution is used as an annotation in the partitioned heritability component of LD score. And the enrichment of heritability explained by SNPs annotated to gene decile is calculated. These methods are obviously related as they use the same data, but they treat it in different ways and so give different answers. So for our purposes, we defined a cell as being enriched, cell type as being enriched for genes associated with intelligence if it was significant in both methods. Skeed and Brewer applied this method to schizophrenia GWAS and identified enrichment in pyramidal cells, particularly the population in the CA1 region of the hippocampus, medium spiny neurons, and interneurons. To test the specificity of this finding to schizophrenia, they tested a number of other phenotypes, including the Sneakers analysis, which showed a generally unremarkable enrichment, although at least one of the methods was significant for serotonergic neurons and pyramidal neurons in the somatosensory cortex. Note, however, that the GWAS, while a similar size to the schizophrenia GWAS, has fewer genome-wide significant loci, which can be shown in this graph here at the top. We thought we could boost the power of the intelligence GWAS by meta-analyzing with a recent study from KCL. Zabano et al. Is a GWAS of very high intelligence individuals from the US population, representing the top 0.0003% of the distribution. These were compared to individuals from the general population in a pseudo-case control study. And the extremely high IQ individuals were hypothesized to be enriched for genetic variants associated with intelligence in the population range. And these are the results from that GWAS, which identified one genome-wide significant locus on chromosome 10. So as such, the aims for our study were to boost the power of the intelligence GWAS by meta-analyzing Sneakers and Zabana, and then to use this to examine tissue and cell specificity across the body and in the brain in particular. In addition to this, we also examined predicted differential gene expression in the brain and sought to understand the sets in which associated genes belonged and to see if those pathways were enriched in particular brain regions. So I'll talk through the results of the study and try to give some methodological data on each part of the results as I do so. So a brief slide on GWAS QC. We combined the summary stats from the two studies. Sneakers is made up of multiple cohorts and they did different levels of QC. So here I've given the minimal levels, but in many cases the minor allele frequency filtered on was more strenuous. So minor allele frequency of 1% rather than 0.1% as is here in UK Biobank. And an imputation info threshold often higher than 0.4, but certain cohorts had that info threshold. In both cases, white Western European ancestry was focused on to avoid population stratification, and the two analyses covaried for various different components. So in order to combine the extreme sample Zabana study with the Sneakers study, we meta-analyzed in metal using the estimated non-centrality parameters from genetic power calculator from each study as the weights. The key assumptions in this method are the variance explained. This is variance of 0.1% is roughly what's observed in other successful GWASs of complex traits, such as the recent PGC depression analysis. And then a further key assumption is the position of the high intelligence cases on the population distribution. I won't dwell on the GWAS 2 because it's since been superseded by larger GWASs, except to say that we identified 25 loci, 12 of which were not seen in the Sneakers analysis, and that 11 of these new loci have since been found to be significant in the recent larger GWASs, although it's worth pointing out that our study, either partially in the case of Davies or wholly in the case of Savage, overlaps with those later studies. So we calculated heritability and partitioned heritability in LD score using the default 53 annotations plus an additional 5 that captured genomic features of interest. And these I've listed on the slide from ATAC sequencing, open chromosome, and that was then combined with the conserved regions of the genome from the standard LD score analyses, and also variants identified in the Neanderthal genome as well. And in both the case of the open chromosome and the Neanderthal genome, we assay both the regions and the regions plus or minus 500 base pairs. So we saw a heritability of 22%, which is concordant with what was seen in the literature. So if I go back to the previous slide, you can see on the right-hand side that similar estimates of heritability captured by common variants are around about 20 with the largest studies. Some of the more recent studies have moved. That's moved up a little bit again to the low to mid 20s rather than 20 itself. But a heritability of 22% is largely consistent with what's observed elsewhere. In terms of the partitioned heritability, the most striking result is enrichment of conserved regions, and this is quite commonly seen in complex trait genetics. And there are a number of other annotations that are enriched and significant, although in these cases each of these is the region plus or minus 500 base pairs, and this makes it difficult to assess the specificity of this enrichment to these annotations. We also performed GWAS analyses using MAGMA, and for these analyses we extended the definition of the gene region from the ensemble coordinates 35 kilobases upstream and 10 kilobases downstream. And the GWAS p-value is then the aggregate of the mean association scores across all SNPs and the top SNP association. Again, I won't dwell too much on this, but we identified a number of novel genes, in part due to that wider gene region definition. And again, most of these have been reported in the largest successor GWASs. Note that the numbers here refer to individual genes. There are several instances, for example, this spire on chromosome 3, where multiple genes are associated at the same locus. So the precise gene enriched is not always clear. So we used our GWAS association study results to assess predicted differential expression in the brain across individuals with different IQs. And to do this, we used the software METREX scan, which I will briefly explain. So with individual level genotype and gene expression data, one can develop a predictive model of gene expression from genetic data. And then in the original PredictScan software, individual genotype data from a separate study could then be input into this model to impute gene expression. And the associations between the predicted gene expression and the outcome can be tested. METREX scan extends this by, instead of using individual level data, allowing summary statistics from GWAS to be input into the model. And then much the same process happens with an imputed transcriptome and a association study on that. So in our case, we identified a number of genes across multiple brain tissues with predicted differences in gene expression correlated with differences in intelligence. To pick out a couple of genes of particular interest, FOXO3, which was a prominent gene both in this analysis and in the Schneeker's analysis, is a transcription factor associated with the process of cell death. This is potentially of interest given recent findings in schizophrenia, which suggest similar processes involved in neural pruning, perhaps. And ring finger protein 123 is a ubiquitin ligase involved in the cellular immune response. Finally, NAGA is a lysosomal enzyme associated with developmental delay and cognitive impairment. So we then performed tissue and cell specificity analyses using the method described earlier, which I include here as a brief refresher. The analyses using tissues across the body from the GTEx repository identified brain as the most enriched tissue, which is reassuring. Within the brain itself, we see enrichment in the frontal cortex, although the distinction here is perhaps less definite than the previous analysis. We then used the KI superset to examine brain cell type specificity. And so a reminder that the level 1 cell types I talk about are these broader definitions, and the level 2 subdivisions are more specifically defined gene cell types. So within the, across the two analyses, we see 6 different cell types that are significant in at least one analysis, of which 3 pyramidal neurons of the somatosensory cortex, pyramidal neurons in the CA1 region of the hippocampus, and embryonic GABAergic neurons are significant in both. As an additional analysis, we then conditioned on each cell type in turn to assess whether the remaining cell types remain significant when the expression in a specific cell type is taken into account. This is done by permuting GWAS Z scores in such a way that the association with the target cell type remains constant. So this mean z-score in each decile is then fixed to what was observed in the real analysis, but others are allowed to vary. And this results in a range of p-values for each cell type controlling for the target cell type. Then if the observed p-value is less than the empirically defined fifth percentile, association is robust to the expression profile of the target cell type. When we did this on our analyses, we found that the observed association CA1 pyramidal neurons was no longer seen when accounting for expression in somatosensory pyramidal neurons. Note that this is the opposite to what was seen in the same analysis in schizophrenia. We additionally examined enrichment in level 2 cell types. There are rather a lot of these, as you can see, so I'll only show those that reach significance in the plots. So on the left, you see the analyses from MAGMA, and on the right, the analyses from LD score. And as you can see, there is a considerable amount of variation between the two methods in this particular instance. So while layer 1 neurons in the CA1 region of the hippocampus were enriched in both, this level of difference between the two methods leads us to not put too much weight on this particular finding. Finally, we examined whether genes associated with intelligence were enriched in biological and disease-related pathways, and additionally added a brain tissue-specific component by weighting pathway membership, which is usually a 0-1 feature of a gene, with the gene's expression specificity in GTEx. Doing this identifies a number of pathways, the majority of which have clear relevance as neural or intelligence-related pathways. And pathway annotations were diverse across brain tissues, with some pathways featuring genes that are broadly expressed, such as educational attainment, which appears in most regions of the brain, whilst others were quite specifically expressed, such as the post-synapse pathway, which is only present in the pituitary gland. Note, however, that these listed pathways are those that pass significance, and that the difference observed are more likely to be a matter of degree of specificity rather than the binary appearance that these data suggest. So to conclude, we found that genes associated with intelligence were enriched at a number of levels, from the brain as a whole down to certain populations of cells, and that sets of genes with like functions that are associated with intelligence may be expressed more specifically in certain brain tissues. Finally, we also suggest that differences in intelligence may be associated with differences in the expression of certain genes within the brain. The results obtained in intelligence had some similarity to those seen in schizophrenia, particularly in pyramidal cell enrichment. The different conditional results, however, are quite interesting. It's unclear whether this is a statistical artifact caused by similarities in the specificity profile of the two populations of pyramidal cells, or whether this might be indicative of more general pyramidal cell involvement rather than specifically one of these two populations. Another alternative is that a third population of pyramidal cells exists but is not assayed within the KI dataset. It's worth noting that since this paper, a similar analysis was performed in Savage et al., and this suggests that some of the differences we observe here may not be as pronounced as they appear. For example, medium spiny neurons, which were not significant in our paper, show enrichment in the larger analysis, and similarly GABAergic neurons, which were significant in our paper, are less significant when with a larger sample. The association with the frontal cortex may indicate that certain cell types, such as pyramidal cells in the cortex, are enriched for genes associated with intelligence, but that others are not, and that this heterogeneity may then hide cell type specificity from observation, which then leads to the question as to whether tissues are too heterogeneous to do this level of analysis, and whether we really need to be looking at the cell types rather than tissues. As mentioned, the important caveat to this is the nature of the cell type specificity information. While the KI dataset is a fantastic resource in terms of its coverage and the methods used, it comes from mice, which, although they show considerable conservation of genes with humans, have obviously taken a different evolutionary path and so have different prominence of and reliance on certain cell types. This may be particularly relevant in intelligence, which is given that intelligence is a fairly specific, fairly characteristic human phenotype. Furthermore, the KI dataset is not a comprehensive coverage of all human cell types available, partly due to the fact that not every cell type is available in mice, partly due to the fact that it's limited to those cell types that are conserved between mice and humans, and limited specifically to the genes that are conserved between mice and humans, but also because it's a growing data resource, and there's still some cell types that need to be assessed and assessed in greater depth. What this ultimately means is that while enriched cell types are likely to be of interest and worthy of further study, non-enriched cell types should not be excluded on the basis of the data alone. As a final comment, intelligence has an interesting and important relationship to intellectual disability. Across the different analyses, we observed a number of genes and regions previously implicated in intellectual disability. I highlight the GMPPB DAG pathway, which is involved in axon guidance and has previously been implicated in muscular dystrophies and myasthenias, which present with mild to severe intellectual disability. This raises the interesting possibility that the same pathways are involved in intelligence across the population range. I note that Lam and colleagues also independently highlighted this pathway in their work, although they focused on DAG rather than GMPPB. However, it should be noted the overall enrichment of ID-associated genes in our work was not beyond that which one might associate by chance. This could simply be a lack of power, and the fact that that association is significant in Lam et al. Suggests that that is a possibility. Or it may be reflective of intellectual disability being both the lower end of the population range of intelligence and being the result of genetic lesions in key genes, which may not be reflected in the population range. So that is my summary of our paper. I'd like to thank the co-authors on this paper, particularly Julian Briouard, Nathan Skeen, and Helena Gaspar. And this was really very much a team effort. In addition, thank you for listening. Thank you.